I am a lucky man because, let's say, every previous presentation gave a bit of introduction to my talk, so I hope it will not take that much time. So anyway, so of course, our reference in this domain of cognitive effect is the Chernobyl data, of course, as presented by Okato, and so it was clearly mentioned that the dose response curve from all this data showed that the most of the effect was at week 8 to 15 of pregnancy, and to lesser extent week 16 to 25, and all lead to mental retardation, microcephaly, and low IQ levels. The new data in this field also is coming from clinical exposure of children showing mental retardation, lower intellectual performance, and stroke. Because children are also important because they are more sensitive than adults, they have higher metabolic activity, and they have a longer life expectancy to develop other cancer or whatever. So yeah, most of my data is based on the Cerebrad project that I coordinated, that ended in 2015, but we continue. So this is about the Cerebrad projects. We continue now, let's say, the study at the different labs, trying to have, let's say, more background in the area. So these are the objective of the Cerebrad projects. I will not go into details. I thank also Florent and Dimitri that they give, let's say, more detail about all the epidemiology. So for the cognitive late effect, we had human data with the hemangioma cohort and also the Chernobyl cleanup worker and in utero exposed. So I will skip again also these slides. And for the cerebrovascular effect, we had also the childhood cancer survivor studies from France, from UK, and from the Netherlands. And so the main conclusions about, let's say, these human data showed that in utero exposed cohort, there is an age-dependent change in cognition. This is, I think, very important, showing that, let's say, the brain is more sensitive during development in utero. So in utero exposed cohort effects are observed below 0.1 gray. In the medical cohorts, so we see effects at 0.1 or 0.054, and in cleanup workers, let's say these are adults, so the dose is like 250 milligray and over, where we start to see some effects. For the cerebrovascular effect, there is a dose-dependent increase in cerebrovascular complication several years after exposure, also in the French and British childhood cancer survivor studies, with an excess odd ratio of 50%. So this data is also in line with the atomic bomb and Mayak cohort. So I will move now more to the animal studies, and there we had designed to study both early events as well as late effects. So, and we choose two animal, let's say two models, irradiation in utero at E11 or irradiation at postnatal stage, which is P10. So we perform a number of behavioral tests. So this is our setup, external irradiation with different doses between 0 and 1 gray. We try to look to neuromotor activity. This is via cage activity, rotor rod gate analysis, exploratory tasks, social exploration, sociability, preference for social novelty, and also learning and memory. So the idea with all these behavioral tests is more to have a functional analysis. We are not comparing, let's say, cognition of a human to animals, but this is more certain, let's say, parameters that help us to understand the effect of radiation. So I will not go into explaining every test here. So what is interesting, there is a change in behavior following exposure to radiation in sociability alterations, impaired spatio-cognitive performance, reduced cage activity, and altered anxiety-related exploration, and defective social memory. So you can see here the graphs showing those dependency or the change in behavior. So this was very important to confirm, let's say, the change in behavior compared to human. So in terms of structural changes, so we perform also MRI to look to the brain structures from different points. So here, let's say the main structural changes, and you can see here the total brain volume that's decreasing with the dose, and this corresponds to what we call microcephaly, and microcephaly it's starting to appear starting from a dose of 0.3 gray. So this is very important. It has been confirmed also in other studies, which is very important. There is an enlargement of the ventricle size. So, and there you can see an increase in ventricle size, which is the most, let's say, significant, is at 1 gray. And also there is a reduction in cortical thickness, which has been shown by François before. So we did also other studies, but this was, let's say, quite old study that we are now trying to revive and increase the statistical power using diffusion tensor imaging, and this is to identify the whole brain connectome and to understand how, let's say, the function of the brain. Yes. So the next step, we tried to correlate all the behavioral tests with the structural changes with MRI. So we find that those dependent change in activity, social behavior, anxiety related to exploration and spatio-cognitive performance in mice, and also microcephaly from 0.33 gray. And also what is interesting, you can see it here, that total brain volume is correlated with, let's say, total beam crossing. This is in the open field, and you can see that the low brain volume corresponding to 1 gray, it has, let's say, the less performance in behavior. So the same here. Also, there is a negative correlation between total brain volume and the latency at day 1. So this is in the Morris water maze. Anyway, here in this table you have all the correlation it established depending on the brain region that is responsible. So in Cerebrad, as I say, we perform also postnatal exposures, so at day 10 after birth. So we can see also here, this is just spontaneous behavior, and we can see a change starting from 0.5 gray. That's the most important. I think it's with 0.5 gray and 1 gray, we see here a deviation in behavior. There is a significant and persistent behavioral deviation in male mice exposed to 0.5 and 1 gray. And also, what was very important is this graph, that external exposure to ionizing radiation with persistent environmental toxicant. So we use different toxicants. These are combined exposure, show different, let's say, dose response curves, and also with different thresholds. This is, I think, one of the most important results of Cerebrad, showing that, let's say, co-exposure, as you know, we are not living also in a pure environment where we are exposed only to radiation, I think this should be translated to human because we are exposed to many other things beside radiation. As a summary of this part, the shape of the dose-response curve for cognitive impairment in animal models shows a linear dose-response curve with age-dependent sensitivity. MRI data showed morphological changes that might be linked with the functional behavioral impairment as shown for other mental disorders as schizophrenia, and postnatal co-exposure with environmental toxicants showed defects at a dose below 0.1 gray. So, but I will skip this statement because this is not the discussion today. So the last part is more about mechanistic. Here I am pleased to present data from our Italian partners in Cerebrad showing, let's say, quite increased apoptosis 24 hours after irradiation, as well as kind of increase in inflammation. So in summary, there is a huge increase in apoptosis, caspase-3, microglial activation with EVA1, and decreased proliferation 24 hours after exposure, and also non-significant reduction in differentiation with SOX2. So here it's more detailed analysis and trying to look at 6 hours after exposure. We can see a very nice linear dose-response curve, let's say, with in terms of G2M cell cycle arrest. So Francois already explained those details. I will not go again in there. So also in terms of apoptosis, a linear kind of linear increase with from 0.1 and 1 gray. So both significant compared to the control. And also we identify defective migration of newly produced neuron. And we can see it here for both 0.1 and 1 gray. So in summary, there is a widespread DNA damage in both low and high dose exposed cortices, a dose-dependent G2M cell cycle arrest, a more extensive and long-lasting apoptosis in the 1 gray irradiated embryo in comparison to the 0.2 gray, 1 gray exposed. And also there is altered DCX expression pattern at 6 hours post-irradiation in both those groups, which is suggestive for a deficient cell migration or aberrant neuron differentiation. So I move quickly to the postnatal irradiation, where also our Italian partners, Simonetta, present here, showed that at P10, irradiation at P10 may alter, let's say, adult neurogenesis, and you can see it with decreased GFAP expression. This is 7 months after exposure, this is very important, and that's what we are trying to confirm, that, let's say, irradiation at early stage can have lasting effects at adult age. So in summary here, there is a decreased proliferation in a dose-dependent reduction in pCNA 7 months post-irradiation, reduction in neuronal differentiation, decreased GFAP level in a dose-dependent manner after 7 months, and also increased neuroinflammation in hippocampus, CD11, also 7th month after 1 gray irradiation. For most, let's say, for prenatal as well as for postnatal, here I am presenting, let's say, a summary slide to not still have a few minutes, okay? Yeah. All the omics data showed, let's say, also changes in gene expression. So as said in the first slide, we tried to look to the early events. This is, of course, just after irradiation, few hours after irradiation, as well as late events, let's say, at adult age, 3 or 6 months after exposure. And so there is still some data to be published, but in summary, here we show this data is just 2 hours after exposure. We found quite a number of p53 dependent genes, which is quite normal. And this is correspond also to the induced apoptosis. But beside that, we identify a group of genes, anyway, that is about 10 genes that are not characterized yet. We are busy with characterizing some of them, and which are all kind of involved in neurogenesis and brain development, which is very important, I think, for the future. It can help in creating kind of biomarkers. So our partner also from Germany, SoilTAPU, also they identified a number of proteins involved in neurogenesis as well as in signaling of other of pathways of axonal guidance, and as well other genes involved in brain connections and network, let's say, functioning. So all these data have been integrated with our, let's say, bioinformatic group that was from Greece, that tried to find, let's say, the most significant pathway to study. Anyway, due to the time limitation, I just summarize here. We have clearly activation of p53 and responsive genes few hours after in utero exposure at E11. Most of these p53 responsive genes are involved in neurogenesis and embryonic brain development, and genes involved in neurogenesis and synaptic plasticity remain also reduced in brain of mice 7 months after exposure to radiation. So the importance also of this workshop is to try to compare to other studies which are not necessarily related to radiation, but for which we observe, let's say, similar effects. Similar effects have been to radiation have been observed for maternal alcohol intake on the neuropsychological development of the offspring known as fetal alcohol spectrum disorders or alcohol-related neurodevelopmental disorder. And there we see quite similar, let's say, pattern in MRI with enlarged ventricles also, and the same, let's say, parameters as we measure with our radiation effects. Maternal immune activation also is an environmental risk factor for brain and behavior change relevant to schizophrenia, causing marked enlargement of the lateral ventricles in adulthood. So, as well as infectious exposure during pregnancy, which is associated with schizophrenia, epilepsy, or autism, and cerebral palsy in the progeny. So, and here, it will come here that the transcriptomic change in prenatal radiation-exposed brain is highly similar to Zika virus infection, and that was mentioned also by François, including induction of p53, the same as we have, and also we compare the expression pattern found by Zika virus and by our, let's say, own expression pattern, and we have a large overlap between the genes induced in Zika virus and the one induced by radiation. You will ask me a bit question how comes, but this is, I think, all stress-related genes that can be, let's say, similar to between radiation and other, let's say, infectious disease. And this is, I think, important to learn from other disciplines for our radiation effects. So I think this is my last. So, no? Yeah, thank you for your attention.